Hello. Today we will talk about the gene expression regulation. It is our lecture number 8. And it is a very interesting question. Let's start with it. How do cells specialize structure and function? Because, of course, you know that all somatic cells, all the somatic cells in every organisms contain the same genetic material. For example, the cells of your eyes contain the same genetic material with the cells in your liver. It is the 100% the same DNA molecules inside the nucleus of these cells. But the question is, how do these cells specialize? Why the cells inside your eyes so different than the cells inside your liver? Uh, Not all genes are expressed in every cell. It is the answer, one of the answers on this question. Because in your eyes, it will be expression of the genes which are very specific in eyes and provide the function of your retina, etc., etc. And there are no expressions of the genes which specific to liver, only to eyes. But in liver, an opposite situation. It will be expression of specific liver genes, the genes which proteins are very common and very useful to the liver of your in your body. But the liver cells uh, shouldn't to express the genes specific to eyes. It is the one answer to these questions about the specific structure and function of the cells with the same genetic material. It is another example, very interesting example. It is the frog, Annabelbs. Their eyes upper half is particularly well suited for aerial vision, and the lower half of their eyes have for aquatic vision. It is very different types of vision. The molecular basis of this specialization has recently been revealed. The cells of these two parts of eye express a slightly different set of genes involved in vision. Even though these two groups of cells are quite similar and contain, of course, identical genomes, but specific gene expression in these different parts of their eyes of this very pretty animal. Okay, let's move on and let's overview the processes of regulation of gene expression. Prokaryotes and eukaryotes alter gene expression in response to the changing in environment, of course. And in multicellular eukaryotes, gene expression regulates development and is responsible for differences in cell types. RNA molecules play many different roles in this process, in the process of regulation of the gene expression in the complex eukaryote organisms. Okay, Let's look more closer in the regulation of on the regulation of gene expression. In this picture, describe the process of differential gene expression by the small regulatory RNA molecule. It is a small molecule because this RNA molecule smaller than this RNA. It is the mRNA in blue, and this is the short or small regulatory RNA molecule. Of course, it is the product of expression of DNA and gene expression too, but it is a special different genes. And after the transcription of these genes, we will have a small regulatory RNA. During the process of transcription, we also can receive the mRNA blue. And it is, for example, it is the gene of, it is a structural gene, and the this mRNA protein coded the some protein. And after the transcription, this mRNA molecule come to the cytoplasm, and during the usual process of translation on this mRNA molecule, ribosomes can to produce polypeptide chains. It is a common way, usual way. But if we will have this short, small RNA, regulatory RNA molecule, this molecule could be complementary to our mRNA. And this is the reason of the process in which our small regulatory RNA sit down to the messenger RNA. And this complex couldn't be translated. Our ribosomes couldn't come, sit down to the mRNA and produce new peptide because of this small regulatory RNA molecule. It is the one of the example of regulatory process in gene expression, in which small regulatory RNA have a specific role. But it's not the all ways, because we also can to observe the regulatory genes working and the specific regulatory sequences in DNA molecule. Let's move on. And now let's talk about the differences in prokaryotic cells and eukaryotes regulation of gene expression. Bacteria often respond to environmental change by regulating transcription too, as the eukaryotic cells. And of course, natural selection has favored bacteria that produce only the products needed by the cell, because if the bacterial cell will produce the proteins which are necessary for this bacterial, it is wasting the resources, wasting the energy, and etc. And only bacterial cells that produce the products which these cells needed, this bacteria will survive, of course. A cell can regulate the production of enzymes by feedback inhibition or by gene regulation. This is the two ways, feedback inhibition and gene regulation. Gene expression in bacteria is controlled by operons model, or the model of operons. And it could be a positive control when the expression provide, or negative control, inhibitory effects. Okay, and let's look here in this picture, which describe the negative feedback, the regulatory in regulation of enzyme activity, and regulation with the enzymes. It is the Escherichia coli, a process of Escherichia coli. You know that Escherichia cells living in the environment of a human colon and depend for its nutrient. These cells depend for its nutrients on the eating habits of the host. If the environment is lacking into amino acid tryptophan, this is the amino acid, which the bacterium needs to survive. Tryptophan is very important for bacterial cells. The cells respond by activating a metabolic pathway that makes tryptophan from another compound. For example, in our environment, for this Escherichia cell, we have no tryptophan. And after that, these bacterial cells start to produce tryptophan from precursors with the help of some enzymes 1, 2, and 3. Of course, these enzymes are coded by specific genes in the bacterial DNA. Okay. Later, if the human host eats a tryptophan-rich meal, for example, some meat, the bacterial cell stops producing tryptophan from these precursors. Thus, avoiding wasting resources to produce a substance readily available in environment, in the colon from surrounding solution. This is just one example of how bacterial turn on and turn off their metabolism to changing environment. It is the negative feedback here. If the tryptophan become in colon in an environment, it will be the signal to stop this process, to stop the producing tryptophan from precursor, and to stop the gene expression, the expression of genes of these enzymes 1, 2, and 3. This is the Escherichia coli regulation of gene expression. And let's look to the basic concept of operons. Operon, it is a cluster of genes. It is the several genes that controls production of a protein product. A cluster of functionally related genes can be under coordinated control by a single on-off switchers. The regulatory switch is a segment of DNA, and it's called the operator. It is the regulatory switch. And usually operator positioned within the promoter. It is the promoter region, promoter for structural genes. It is the structural genes one, two, three structurals, and it means that these genes encode the proteins, which is the structure of genes. This is the operator, this is the promoter region to the transcription of these structural genes. But we also have these two genes, the regulatory gene and promoter for regulatory gene. And operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. These two genes are needed to controlling of these genes. Okay. The operon can be switched off by a protein repressor. Let's look here in this picture. This is the picture again about the Escherichia coli and tryptophan metabolism and all tryptophan producing. The operon can be switched off by a protein repressor. This is a repressor, a red in this picture. The repressor prevents gene transcription by binding to the operator and blocking RNA polymerase. Let's look here on the top of this picture. It is the gene. It is the fragment of DNA molecule. We have a regulatory gene, promoter of this gene, and the regulatory gene. The product of this regulatory gene, it is the our repressor, the protein. But usually this repressor is in inactive state. When in the cell is lack of tryptophan, without tryptophan, this repressor will be inactive, in inactive state. And when it's so, it's true, the repressor couldn't be bind with the operator. As you see here, the operator has a specific structural parts to their this protein, to the repressor. But inactive repressor couldn't bind with these specific parts of the gene in operator because in inactive state, a repressor has another form, different form. But when tryptophan, it is the co-repressor, when tryptophan comes, our repressor become active in active form. And when it's in inactive form, now this protein could bind to the operator and repress the expression of these genes, these structural genes, which encode our subunits for tryptophan synthesis. So, if in Escherichia coli we have no tryptophan, Escherichia coli wants to receive it, and when it's so, our repressor in inactive form, and it couldn't bind here to the operator, and this is the reason that operator have no inhibition effect, and our RNA polymerase can bind to the promoter, and the expression of these genes, the genes which needed to polypeptide subunits to tryptophan synthesis, this expression can be in the cell. And when in cell we have enough tryptophan molecules, these molecules of tryptophan will be bind to our repressor, and the repressor become active and bind to the operator, and in this position polymerase couldn't bind to the promoter region, and this is the reason of stop transcription here. And of course, if polymerase couldn't work, we have no this messenger RNA for these subunits, and of course we have no any synthesis of tryptophan, because we have tryptophan, enough tryptophan molecules in our cell, in our Escherichia coli cells. This is the general principle of operon model. Okay. Eukaryotic cells. What about the eukaryotic cells? Eukaryotic gene expression is regulated at many stages. All organisms must regulate which genes are expressed at any given time, because the environment very different in different time. In multicellular organisms, regulation of gene expression is essential for cell specialization. As we said in the beginning of our lecture, remember their frog with the different parts of their eyes, or for example, the cells of your eyes and your liver cells. It is very different gene expression inside the same cells, the cells same in DNA which they contain. Almost all the cells in an organism are genetically identical, except, of course, gametes. You know it. Differences between cell types result from these different gene expression patterns, and the expression of different genes by the cells with the same genome, as we said. But abnormalities, maybe some kind of dysregulation in gene expression can lead to diseases, including cancer. And so in eukaryotic cells, we could observe gene expression regulation at many stages. Let's look here in this picture. It is the stages in gene expression that can be regulated in our eukaryotic cells. For example, this is the nucleus. This is big area. It is the nucleus. Nucleus with the DNA molecule which form chromatin. Chromatin, it is the DNA plus specific protein histones. And chromatin could be modified. We will talk about it a little bit later, about chromatin modification. It is the packing or unpacking DNA. And in this process, histone acetylation, histone methylation, etc. processes are involved. So we could regulate gene expression on this level, on the level of chromatin. But also we can regulate the gene expression during the transcription stages. It is another level. If in this level we will switch on or switch off some genes because of these processes of chemical modification of histones, acetylation of histones, methylation or demethylation of histones. And this chemical modification will be switch some genes because, for example, histone acetylation make our DNA molecule unpacked. And in this state, our DNA more available to transcription machinery, to the specific enzymes, polymerases, which produce the transcription. But in these stages, during the transcription, we can do some regulatory effect. For example, in eukaryotic cells, we can to activate transcription or inhibit transcription. In some cases, the transcription of some genes can be more active, and as a result, we will have more proteins molecule. But in some cases, with the help of specific factors, the specific enzymes, the transcription of some genes can be not so active as usual, and we have less proteins as a result. But another level of transcription regulation is RNA processing. In eukaryotic cells, as you remember, of course, we will have a primary transcript at the first time, but after RNA processing, we will have a specific messenger RNA, which have to be shortened and receive the specific parts on the ends, a cap, and poly-A tails. This is the mRNA in the nucleus, and we could do different processing, different ways of processing in this level. For example, the alternative splicing. We will talk about it today a little bit later. The next level, it is the regulation on the level of transport to the cytoplasm. This messenger RNA molecule have to be transported here from the nucleus to the cytoplasm, because only in the cytoplasm we have the ribosomes. And maybe in some cases our cell have not enough transport proteins, and these transport proteins very inactive, and our messenger RNA will be degrade inside the nucleus or even stay here in nucleus, but we have to have this mRNA in cytoplasm. But without the help of specific transport proteins, our mRNA stay here in nucleus. And of course, this is the reason of lack of protein molecules here in the cytoplasm. Okay, but in cytoplasm, on the level of translation, we also can to alter our gene expression, because with the help of, for example, some small RNA molecule, as we said at the first part of our lecture, with the help of this small RNA molecule, our messenger RNA can be degraded. And of course, this is the reason of the protein absence in this cell. After that, on the level of polypeptide chain construction, it will be a regulation too. Because during the protein processing, when we have to receive active protein from polypeptide chain, it may be some chemical modification, and we will receive another protein, the protein with another shape. And of course, if the structure of protein is changed, the function will be changed too. And also the transport to cellular destination, it is the another level of regulation of gene expression, because after the producing of protein on ribosomes, these proteins have to be destined to the specific parts of the cell. For example, some proteins some proteins are very useful to axons of the nervous cells, and they have to be destined here in the axons. And axon of the cell can be very long, maybe some centimeters in, for example, some mollusks. And these proteins, which are necessary to the, for example, synaptic transmission, these proteins have to be destined here from very long distance, of course, with the help of some enzymes. And during this transport process, we can to regulate, of course, this destination. For example, if we have not enough enzymes, transport enzymes, our axon will have a deficit for some proteins. And the last level, it is the level of degradation of protein. After all of these processes, all of these stages, we could receive a good protein, active protein, but by some reasons, these proteins could be degraded. And we will have not a good active protein, but the amino acids, the blocks, the units of this polymer. And of course, in this case, we have no active protein and the lack of function of this protein. This is the stages in gene expression that can be regulated in eukaryotic cells. So let's look more closer on this level about the chromatin regulation, about the regulation of chromatin structure. As we said, genes within highly packed stages are usually not expressed. If our DNA very packed, the polymerase, the transcription machinery couldn't sit down here on the DNA molecule. On this picture, blue, it is the DNA. These blue lines, it is the DNA which wound around the histones. These parts, it is the histones, it is the specific protein. Histones with the tails. Every histone have a tail. And DNA around these histones. And if our DNA very packed, this state we called heterochromatin. Heterochromatin. And heterochromatin usually not expressed. If our DNA unpacked, in this case, we will see that our DNA fragments of DNA are open, and polymerase and transcription machinery could sit down here and make a messenger RNA. But here, polymerase couldn't do it. This stage of active unpacked chromatin, it is the euchromatin. 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 And it is the expression. It's switched on of expression. And the chemical modification of histones and DNA of chromatin influence both chromatin structure and gene expression. What kind of chemical modification? For example, it could be acetylation. Here we could see the unacetylated histones. Here on the tails of our histones, we have no any acetyl groups. It is the free histone tails. And this is the reason of packaging of chromatin. It is the heterochromatin and no expression in this case. But here, after the specific chemical modification of our histones, it is the acetylation. Here it is the acetylic groups on the histone tails. And this is the reason, the acetylation, it is the reason of unpackaging of chromatin. And this is the euchromatin, and expression, gene expression can be here. Histone acetylation, it's not the one in the modification of chromatin. Also, it may be the addition of methyl groups, methylation, or phosphate groups, phosphorylation. But in histone acetylation, acetyl groups are attached to positive charged histone tails. And it leads to unpackaging because it changes the charges of histones and our attraction of the forces of attraction of DNA, which have DNA, we have negative charges, and histones with the acetyl groups became more negative too, and this is the reason of repel of our DNA molecule and histones, and our chromatin will be open, and after that the expression can occur. This loosens chromatin structure. This process loosens chromatin structure. But the addition of another groups, for example, methylation or phosphorylation, have another effect. For example, after some modification, chromatin can become more more packet and heterochromatin. It depends of the chemical group and the position in which this chemical group will be attached here to the histone tails. Okay, it was the simple model of histone tails and histone modifications. And the histone code hypothesis exists. And this hypothesis proposed that a specific combination of modifications as well as the order in which they occur help determine chromatin configuration and of course it influenced to transcription. And as in the case of genetic code, which was open or maybe hacked in 20th century, we have to crack histone code maybe in during the 21st century. It is a very important area of biology which called epigenetics. It is the part of the genetics in which we study not the mutations but the modification of gene expression. Okay. Except the modification of histones, the molecule of DNA could be modified too. Not the histones, but the DNA. And DNA could be methylated. DNA methylation, the addition of methyl groups to certain bases in DNA. Usually it is the C PG dinucleotide. To the C, we could to attach a methyl group. DNA methylation can cause long-term inactivation of gene in cellular differentiation. For example, if we attach some methyl groups here in the DNA, it means that this gene, the genes which are positioned here, couldn't be transcription. And there is no transcription due to methylation of this part of DNA molecule. And for example, during the cellular differentiation, when the eye cells became eye cells and liver cells became liver cells from the precursor cells, from the stem cells. And it is very long-term inactivation of the genes which not specific to this organ, not specific to this tissue. For example, in the eye cells, the liver genes will be long-term inactivate due to DNA methylation of these genes. This is the the general shape of epigenetic regulation of cellular differentiation in very, very common way. And this picture is very interesting because it describes the factors which could to affect epigenetic modification of DNA molecule and chromatin. And as you see, it is very spread, very huge set of factors. Our diet, what you eat, it can modify your DNA and your chromatin, and this is the reason that diet can affect uh, our state and our health, because, for example, the presence or absence of some nutrients could methylate or unmethylate your DNA, and some genes will be switched on and some genes will be switched off. It's very interesting. Seasonal correlation, seasonal conditions, uh, some viruses, some toxic chemicals, drugs of abuse, uh, even finance status. Have you enough? Do you have enough money or not enough? And this is the reason of your chemical modification of your genome. Exercise, of course, the sport, microbiome. Microbiome, it is the modern topic, hot topic in the biomedicine, because today we understand that microorganisms which habit here in our intestine uh, can to produce some nutrients which can modify our DNA and histones. Drugs, uh, even alternative medicine, maybe the epigenetic, it is one of the ways, one of the reason of action of some alternative medicine tools. Social interactions, and psychological state, your emotions, your anxiety, depression, etc., etc. All of these factors could alter your epigenetic modification and so alter your genetic transcription. It is very hot topic in biomedicine. And as an illustration of this influence of environment through the epigenetic to the behavior and the functioning of organism, I want to say about the experiment with the rats. And it was the experiment in which stress in early life of these pups become the reason of genetic alteration in their adults animal. In this case, we have two strains of rats, one strain and another strain. This strain number one, it is very attentive mothers. It is the genetic selective, the result of selection in the lab. This mothers was very attentive, and they uh, uh, grooming their pups and provide enough milk for them, etc., etc. But another strain, it was the inattentive mothers, bad mothers. Their children have not know enough eat enough meat, enough food, sorry, and enough attention. And after that, when these animals became adult organisms, the researchers provide stress to these animals and research their reaction to the stress, their behavioral reaction to the stress. And these animals the children of good mothers was more stress resistance when he became adult. And these animals, the children of bad mothers, was less stress resistance. And after these behavioral experiments, the researchers uh, cut the hippocampus. Hippocampus, it is the structure inside the brain of these animals. You have hippocampus too. The main function of hippocampus, it is the memory and the regulation of stress reaction. And of course, in this hippocampus, our animals have differences too. In the case of good mothers' children, it was the rarely methylated genes. In case of bad mothers' children, it was the genes highly methylated. And as you remember from our previous part, when our DNA have a methyl group, it will be inactive DNA, no expression, no gene expression. And so we could conclude that in case of prenatal, or not prenatal, but very early stress in these animals, we will receive animals with the inactive of some genes in the hippocampus of this animal. But it's not enough to explain. And another experiment of these researchers was to exchange of children, of pups here. Maybe these differences in behavior and gene expression in hippocampus due to the genetic reasons, because this is the same in genetic material, are not about the stress, the behavior of the mother. And in this experiment, we change the children, and mothers raise foster pups. But after the same experiment, after the same evaluation, we had the same results. In this case, when genetically it was from here, from these good moms, but they arises with the bad moms. Our animals in adult was less stress resistance and highly methylated genes, have highly methylated genes in hippocampus. In this case, we had the genetically animals from bad mothers, but they have a good childhood, and this is the reason of more stress resistance and the more active genes, because genes are rarely methylated in hippocampus in this case. It's very interesting results, and these results describe the old psychological statement that all our problems, all our troubles in adulthood have roots in our childhood. On our problems come from childhood. Okay, but it is very interesting to understand what kind of specific genes was methylated or unmethylated. But at the first time, we have to make another experiment, the last experiment, when we took the inattentive mothers bred the pups of these animals and inject in the brain, in the hippocampus of these pups, specific substance, trichostatin A. This substance can remove methyl groups, and so we try to change epigenetic modification. We want to unmethylate DNA in these animals, the animals which was exposed to very early stress factor. And after that, we will research the epigenetic changes, and of course, and so methyl groups was arise in this case in hippocampus, and the behavior of our animals become a normal behavior. Okay, but when their researchers look into the particular genes which was hypermethylated in this case, they conclude that it was the specific genes in hippocampus which encode the GR receptors. It is the glucocorticoids receptors, the receptors to the hormones of the stress. This is the axis of the stress hormone producing. We will talk about it a little bit later when we will talk about the human physiology. But now we have to understand that these genes, the genes of receptors to the glucocorticoids, to the stress hormones, was methylated in their children of the bad mothers. And this is the reason of the less quantity of receptors in the brains of these animals. Because, of course, again, if this DNA here, in this DNA will be methylated, this is the methyl groups, in the promoter of the gene of GR receptor, the specific transcription factors couldn't sit down here, and our expression of the genes of these receptors is inhibited. And of course, it decreased the GR expression, and not only in early life but in adulthood we will observe the same picture. And this is the case of bad childhood of rats, not only the rats but human too, because we also have a hippocampus and our stress hormones are very same with the rats. But if their childhood was happy, if your mother was attentively, then in your hippocampus it will be no methylation group here in the promoter of these genes, and our transcription factor can sit down and provide our transcription. And it increased GR expression in early life and in adulthood. And these receptors very necessary to the regulation of this axis of these organs and pathway. This is the explanation of how epigenetic works. It's a very interesting topic, I think. Okay, let's move on and let's talk about the regulation of transcription initiation. Chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind the transcription machinery. For example, let's look here in this picture. In this picture, we could see the specific region which have a name enhancer. Associated with the most eukaryotic genes are multiple control elements. Control elements, it is the segments of non-coding DNA, the part of DNA that do not code the protein. It is the non-coding DNA that serve as binding sites for transcription factors. Transcription factors, it is the protein that help regulate transcription. And control elements and the transcription factors, they bind critical to regulation of gene expression in all cell types. They initiate transcription in eukaryotic RNA polymerase requires two assistance of proteins that cause transcription factors. Here in this picture, we could see not only the region of enhancer. Enhancer, it is the part of DNA molecule which could bind with the specific agent activators. Activators proteins bind to this control element, distal control element enhancer. This enhancer, on this picture, it is the 3 sites, 1, 2, 3 sites to the binding of activators. And also we have a structural gene here and the promoter of this gene. Theta box, you know that this is the part of the promoter in which our polymerase molecule should bind and provide the transcription. Let's look here in the point 2, and here we could see that our DNA molecule change the shape. It's not a linear form because here we have DNA binding protein, the specific protein that brings the bound activators closer to the promoter. Here, all of 3 activators bind with the enhancers, and it became more closer to the promoter region here because of this protein which bent our DNA molecule. And after that, activators bind to the mediators. These are different protein subunits. It is the mediator proteins. And the activators bind here with these proteins and with the transcription factors, which help to polymerase. And after that, our polymerase can bind with the promoter region and it will be RNA synthesis. This is the example of the model for action of enhancers and transcription activators, and it is again the regulation of gene expression, the example of the regulation of gene expression. And here it is the cell type specific transcription. Here we could see two type of cells. It is the cells in your liver and a lens cell in your eyes. It is the gene of albumin, which is needed to these cells, the lens cells. Oh, sorry, not here, but of course, crystalline. It is the protein which needed to the lens cells, and albumin for liver cells. And here in the liver cell, we can see that, of course, these cells, the nucleus of these cells, have as needed albumin gene and not needed crystalline gene. But crystalline gene is not expressed, but albumin gene expressed. Why? Because of transcription factors and activators. In the liver cell, we have a specific for liver cell activators. And these activators bind to the enhancers here only in albumin gene. Because here in crystalline, it is the different activator. And maybe in the liver cell we have the activator 1 but have no 2 and 3. And one activator, it's not enough to produce all of these changes. And only genes which is needed will be expressed here. The same here in the lens cell nucleus. Again, two genes, albumin and crystalline, but a crystalline gene expressed and here not expressed. These genes not expressed. Why? Because of activators. Here in lens cell, we have specific activators, only specific to this gene. But we have not enough activators to albumin gene. And this is the reason of type-specific transcription. Okay, let's move on. And some words about the splicing. It is the post-transcriptional regulation, that mechanisms of regulation after transcription. Regulatory mechanisms can operate at various stages after transcription, and such mechanisms allow a cell to fine-tune gene expression rapidly in response to environment changes. And for example, it is the alternative splicing. Here in this picture, it is alternative splicing. Different mRNA molecules can produced from the same primary transcript. The same primary transcript here from the same gene in DNA, and it could be different, two different or more than two different mRNA molecules. Of course, it will be different proteins as a result. Why? Because of exons and introns. In our previous lecture, we discussed it. We have not only exons, coding parts of gene, these 5 genes, but introns, and these introns we have to cut after the processing during the splicing. And after this cutting, our exons have to be fused, linked to get to each other. But in some cases, we can link this node of these exons. For example, in this case, we missed fourth exons, but in this case, we miss 3 exons. The exons are number 3, and this is the reason of different mRNA molecule. And our proteins, of course, it will be different proteins. This and the alternative splicing. Okay, and their regulation of gene expression by microRNA, the short RNA molecule. This is the same mechanism as we said, mentioned in our first part of our lecture, but in very briefly, look here in this picture, and here we could see that microRNA, it is the short molecule of RNA, with the specific, with the complex, with the specific protein complexes, could to be the reason of degradation of messenger RNA because of complementarity, the nucleotides in microRNA and in messenger RNA. And this is the reason of degradation of messenger RNA. Or in some cases, maybe our miRNA molecule not the complementary to the all messenger RNA, but in only partly, and this is the reason of transcription will be blocked. And this is the reason of absence of proteins. It is the another very tuned and active mechanism of regulation of gene expression. Okay. And some words about the transcripts of eukaryotic genes. I want to remind you that the transcript, the major messenger RNA, have only exons, here only exons, not introns, because introns was cut, and of course some specific region, a cap with phosphate groups, and polyA tail, and some untranslated region here. It is the shape of major messenger RNA. And some words at the end, some words about the cancer. Very briefly, but very important to understand that the cancer, it is the result of the genetic changes in the changes of gene regulation of some genes. For example, the one case, it may be a translocation, and transposition. It is a mutation when a gene moved to new locus, and new locus is under the new control element, the new promoter. And this is the reason of producing extra proteins and extra quantity of the proteins of growth factors, growth stimulating proteins. And these proteins start to stimulate the growth and development of our some tumors. But it's not a one mutation, one kind of mutation. In some cases, it may be gene amplification. Gene amplification, it means multiple copies of the same genes. There was one copy in the physiological state, but after this mutation, after amplification, it could be several copies of this gene. And of course, this is the reason of increasing of growth factors and increasing of growth in some cells. And point mutations. Point mutation in oncogenes, the genes which encode the proteins specific to growth and development and cell cycle proteins. In this case, it may be point mutation in the promoter regions or another control element, and this it increase transcription and translation, and it will be excess of growth stimulated protein. In some cases, it may be a point mutation inside the body of the genes, inside the exons, and it could be the reason of hyperactive or degradation resistant protein. And all of these cases, it may be the reason of cancer. Cancer, it's not a result of one mutation, but it is the result of several mutations and an epigenetic mistakes, the epigenetic alteration in gene regulation. And the last picture is about the cancer again. It is the an example of colon cancer. And it is the multi-step model for the development of this cancer. And all of the cancers are multi-step. First of all, we have loss of tumor suppressor gene, for example, the gene APC in this case. And this is the reason of growth and development small polyp. It's not a cancer in this case. It's not yet a cancer. It is the polyp. It is the tumor, but it is the good tumor. After the next mutation, we observe the activation of some oncogene, for example, RES oncogene. And after that, loss of tumor suppressor gene, the gene which encode suppressor proteins, suppressor for growth and development. And our polyp became larger. If there was another mutation, for example, loss of tumor suppression gene P53 and some additional mutation, we will receive a malignant tumor, a carcinoma. Here it was an adenoma and carcinoma in this case. There are several steps in this cancerogenesis processes. Okay, this is the end of our lecture about the gene regulation processes. Thank you for your attention.